Happy Friday, everybody. This is the first of our free response Fridays. The first question we're going to look at is going to be a 10-point question. I'm going to try to tailor all the questions to the question types you'll probably see when you get that exam on May 18th at 2 p.m. So please make sure that you take a look at that. This question is from the 2017 AP exam. It was question number 2. And so if you want to look it up online as we're doing this, you can kind of go back and forth, open up a new window on your computer, whatever the case may be. I do not have the questions up. I only have the question prompt up. And you can see it's a pretty lengthy question. And when you look at it already, there's a graph. That's the first thing you give you. There's your line graph. And then they give you this data table at the bottom with controls and something called KR and TMB. And if you go ahead and read here, you are going to see these very, very large words. Carrageenans and trimethylbutanoids. Here's the thing. I know you guys get caught up in, I don't know what those things are. I don't know what they are. I don't know what they do. I can't understand them, okay? Don't worry about the names. Again, call them puppy dogs, call them kitty cats. I don't care what you call them. But the K's and the T's. That's what we're going to call them. All you need to know, though, by looking at this picture is they are water-soluble compounds. They are found in smoke, and they are deposited as a result of fire. Okay, so whenever a fire happens, these chemicals, okay, dissolve in the water and they seep into the soil. And then in the subsequent generations after that, what you're going to see is you're going to see that they're going to have effect on plant growth. But here are the questions. And I'm not going to be able to put up a picture of them on the system I'm using, but I will read you the question. And if you want to look up the question, again, just Google 2017 FRQ AP Biology. It's question number 2. Question letter A says, the researchers made the following claims about the effect of KAR and the effect of TMB on seed germination relative to the control treatment. So again, they're saying relative to the control. And so we're seeing how KAR and TMB, how they regulate, okay, or the effects of them on seed germination relative to the control treatment. And so they want you to determine how do these things compare to what would normally happen with just water, because that's probably our control group. And so the first claim they make, the first claim says that, and this is written on the question, KAR alone affects timing of seed germination. And I'm just going to seed germ. Okay, that was their first claim. The second claim they made is that KAR alone affects percent of seeds germinated. The third thing that they said was that TMB alone affects the timing of seed germination. And then you can probably guess the last one. They said that TMB alone affects percent of seed germination. Now, here's the thing. It says after these 4 bullet points in question A, it says provide support. And those words are in bold. So provide support. Okay. Using data from figure 1. Now, I didn't even read the question yet. I didn't even go through and read any of this over here. Okay. I don't need to. The first thing you should do when you get a free response question is to look at the questions. Don't read all the fluff that comes before it, because you might not even need it to answer a question. Okay, we found that when we did the mock exam. I think there was something about what makes up a nucleotide. Okay, well, you didn't even have to read to figure out what makes up a nucleotide. You just needed to know it. But it says here, refer to figure 1. So only using stuff from figure 1. So for part A of this question, it's 4 points because you have to support all 4 bullet points. don't even look at anything past figure 1. Don't worry about it. You are only supposed to use figure 1 to support these claims. All right, so let's go ahead and do that. Let's look at the first bullet point. So if I was going to go answer this, I would look at this and I would say, okay, KAR alone affects the timing of seed germination. So I come up here to figure 1 and I'd say, okay, here's time. Let's see the time at which seeds start to germinate as with KAR as compared to the control. Okay, well, control, that starts germinating about here. That's when we start to see actual seeds germinate. KAR starts here. So what would I say? Well, I would say KAR alone, it is going to cause seeds to germinate sooner. Or you could say even quicker. Or you could say faster. Whatever you want to say. But as compared, I'm going to move this up if I can. I don't know if I can. As compared to the control. Okay, so there you go. That's the first bullet point. We answered that. KAR alone affects timing of seed germination. Yes, it does. It causes the seeds to germinate quicker. The second part of this, my next sentence, and again, each point should be at least one sentence, and you have to write in full sentences. My next point would be about KAR alone and how it affects percent. All right, well, let's take a look. Here's control. And this doesn't have anything to do with how fast they get to that percent. It just has to do with the percent. Well, what is KAR? What do we see about percentage-wise in seed germination versus control? Well, we see KAR alone causes the percent of seeds, and you would write out percent, seeds germinated to increase compared to the control. All right, that's part number 2. Now we've answered 2 points, okay? Next, TMB alone. How does it affect seed germination? All right, so TMB alone, and let's take a look at what it does compared to control. So the TMB alone, if we look up here, here's our TMB down here. Okay, here's our control up here because we're comparing it to the control. The TMB alone, it looks like, causes the seeds to germinate slower, okay, or they won't germinate as quickly. And so you write TMB alone causes seeds to germinate slower than the control. And lastly, the TMB, and we're looking at how does it affect the percent of seed germination. So again, we go back up here, we look at our percent of seed germination. Take a look. Ooh, only 10% of seeds germinate with TMB alone. That's not good. And so TMB alone causes less seeds to germinate than the control. Done. Okay, that's 4 points right there. I used 4 sentences, 4 points. Part A is done. I provided my reasoning. My reasoning was, if you take a look at the graph, you'll notice there it is. Okay, that was the support that they wanted you to figure out. All right, guys. Part B of this question. So that's 4 points. Part B says, make a claim about the effect of rinsing on the binding of KAR to receptors in the seed, and about the effect of rinsing on the binding of TMB to the receptors. Identify the appropriate treatment group and results from the table that when compared to the controls, and provide support for each claim. Okay, so this one is a little bit more difficult. So you got to kind of break down the question here. And so I want you to make a claim about the effect of rinsing. So basically it says, how does rinsing affect the binding of KAR to the receptors on a seed. And so again, this is a chemical. It's going to bind to the seed receptors. And as soon as it binds to a receptor, it's going to cause germination or not germination, depending. And so it wants to know, how does rinsing affect the binding of KAR? And, okay, that's 1 point. And it wants to know, how does it affect the binding of TMB? So that's another point. And it says, identify the appropriate treatment and results from the table that, when compared with the controls, provide support for each claim. And so you're going to say how the rinsing affects it, and then you're going to look at the table. So they're talking about this table here, and you're going to provide which treatment supports that, supports your claim. Okay, and so you're going to have one treatment. Okay, you're going to have support here for this, and you're going to have one here for support for this. And so you're going to have 1 point to say how rinsing affects the binding of KR, 1 point for how rinsing affects TMB. Okay, and when they're talking about rinsing, they're talking about rinsing with water. And then they want you to give you support by using this table below. So what is the support for your claim here, and what is the support for your claim here? So overall, part B is going to be worth 4 points as well. Okay, so let's look at that data table down here. And it's showing you the control results. It's showing you when it is treated here with KAR. Okay, and then water is not present. Here they're showing you water being present, and they're giving you a control result, okay, which is here. That's the control result. When treated with KAR, okay, and rinsing, they're giving you different from control. And if you look at your TMB alone with rinsing, it's the same as the control is the result, okay, because they said with rinsing, okay, the effect of rinsing. And so the rinsing right here is the water. That's what the water rinse is. And it tells you that, effective chemical treatment and water rinse. So they didn't do water rinse on the first three. And so I would guarantee you're probably not going to look at the first three treatment groups here. You're going to be look at the treatment groups that use the rinse water and say how rinsing affected your control result. All right, so if you look at, I'm just going to erase this since there's a lot on here. All right, so when you get done here, what you're going to realize is that you are making those 4 points by stating binding is not affected by the water rinse. That was your claim. Then you gave support to show why you claim that. Same thing for TMB. TMB is affected by the water rinse, and then you showed me why you made that claim. When you make a claim, making a claim means you have to say that something happens, a yes or a no. In this case, it asks, make a claim about the effect of rinsing on the binding of KAR to the receptor. Okay, binding of KAR to the receptor site. I made a claim that it is not affected. That's my claim. For TMB, I said it was affected. So making a claim is a yes or a no. Yes, it does happen. No, it does not happen. And then you have to support that by using the data table. Okay, that's what I ask you to do all the time in science. We do an experiment. You give me the results, and I say, why do you think that happened? And then you make your claim, and then you support it. You support it by using the data that you have. And data is usually found either in a graph or a data table. So whenever you have to make a claim and support it, look at data tables and graphs, because that's where you're going to find most of your support. All right, so that's 8 out of 10 points that we covered in parts A and B. In part number C, the only thing that's left are 2 points. It says, there is an intense competition by plants to successfully colonize areas that have been recently cleared by a fire. Describe one advantage of KR regulation and one advantage of TMB regulation to the plants that live in an ecosystem. All right, so first of all, we need one advantage of KAR, and then we need one advantage of TMB. B. Okay. Notice, guys, I haven't even really read any of this right here. I mean, it's okay if you did. It might be a little bit confusing, but I really haven't looked over that a whole lot. And so I do know that KR and TMB, I did read, of course, in the first part, is that they are water-soluble compounds found in smoke that are deposited in the soil as a result of a fire. And so when a fire happens, these things are going to be deposited in the soil. And so the question is, in part C, well, there's an intense competition by plants to successfully colonize after a fire. Okay, so we know after a fire, KAR and TMB are both going to be in the soil. So what's an advantage of having KAR if you are a plant? What's the advantage of that? And so take a look again at your graph. What would be the advantage of having that KAR? Okay, and there's a couple things you could go with here. First of all, what I realize is that germination occurs quickly when other plants are not using resources. And so if you can germinate sooner, then you can get those resources before everybody else. Okay, so with KAR, germination occurs sooner. You'd be able to get those resources earlier. Okay. Another thing you could say is that they would be able to germinate sooner so that they can compete, okay, with the resources that are available. Something like that would probably be your best bet. And what would be the advantage to being KAR, or excuse me, of TMB? TMB regulation is an advantage for plants in dry environments because they can stay dormant longer. Okay, they can stay dormant longer, which is a good thing for these plants. If you have a plant that has TMB, they're not going to, you know, germinate as quickly, and therefore if there's not enough water available for germination, you know, they can wait it out a little bit longer. And so being able to wait out until there is enough water is actually a good thing to outcompete, because those that grow quickly don't have enough water, they're going to die as soon as they germinate. These seeds hold out until there is water, they'll be able to do it. Okay, so those are your 2 points for that part. One advantage of KR and TMB. KR advantage I think was easier just because you could say easily that they start growing quicker and therefore we'll get the resources quicker. TMB regulation, you might have had to think about that one. The dormancy part, we don't think of seeds being dormant. But when you buy a pack of seeds at the store, those seeds are dormant. Okay, when you put them in the ground and you give them water, they grow a plant. And so keep in mind, seeds are dormant until we give them some type of water or chemical. In this case, the chemical was KR TMB, and then we added that water rinse to it. So do keep that in mind. All right, so the first FRQ wasn't too bad. There was a lot of information there, a lot of words you didn't know, but like I said, read the questions first. You might be able to answer stuff easily without having to do any reading. And that's why I like this question. You didn't even have to read to get the first 4 points. You just had to look at the graph. Okay. All right. I'll be back next Friday with another 10-point question, and it'll probably have something to do with experimental design. So we'll see you then. Bye.